During this video, we will investigate how to use Punnett squares to solve genetics problems. We will look at the three types of dominance: complete dominance, incomplete dominance, and codominance. When solving genetics problems, there are a few steps you should follow. The first one is to set up a key of alleles. So, look at your problem and define which letter you will use for the dominant allele and which letter you will use for your recessive allele. The second step is to state your cross. So look at your parents and determine what the genotypes of each parent are. The third step is to put the gametes from the parents on the sides of your Punnett square. Once you have the gametes set up, you can fill in the offspring genotypes inside the squares of your Punnett square. The last step is to go back and answer the question. Be sure that you look at the actual question and answer what is asked. This is the most missed step by students whenever they are solving genetics problems. So far, all of the problems that we have been working on have been with complete dominance. This means that the dominant allele completely masks or hides the recessive allele. Let's work through another example. Here is our next problem. Brown fur is dominant to white fur in rabbits. An individual with a homozygous dominant genotype is crossed with a recessive white individual. What is the predicted genotypic ratio of the offspring? What is the predicted phenotypic ratio of the offspring? So this first problem is a case of complete dominance. Our first step is to make ourselves a key of alleles. So brown is dominant, so big B will represent our brown allele. White is recessive, so our lowercase b will be the white allele. Okay, so next thing we need to determine is our actual cross. So blank times blank. What are our parents? So an individual with homozygous dominant genotype. So that means that homozygous is two of the same alleles, and we're talking about the dominant allele. So the first parent is big B, big B. Okay, the recessive allele is the little b, so a white individual is going to have two copies of the recessive allele, and that's the only way to get that white coloration to show up. Okay, so once we have our cross, we're going to divide or segregate our gametes and put them on a Punnett square. So we'll draw our Punnett square. Okay, so first parent is big B, so we're going to put a big B here and a big B there. Second parent is little b, so we'll put those gametes on the top of our square. Once we have that, we're going to fill in our square. Okay. So one gamete from each parent. And then we have to go back to the problem and answer what is actually asked. So this problem wants to know the genotypic ratio, which is what letters are actually in our box. So the genotypic ratio is 100% big B, little b, or heterozygous. So the phenotypic ratio is going to be a translation of our genotypic ratio. Okay, so what color are these rabbits? So big B, heterozygote has one dominant allele, which means that all of these rabbits are going to be brown. Here's another practice problem. What is the possibility that a cross between a pigmented giraffe, who is homozygous dominant, and an albino giraffe, homozygous recessive, will yield albino offspring? This second problem is very similar to the first. So we're talking about pigmentation versus albino. Okay, so a pigmented giraffe, let's use big A for pigmentation. And we'll use little a for albino. So albino is the recessive allele or a recessive trait. Okay, so our parents We set up our cross. We have a homozygous dominant giraffe, big A, crossed with an albino giraffe. And the only way to see the albino allele show up is to have two copies of the recessive, so little a, little a. So put those gametes on our Punnett square. First parent is big A, second parent is little a, little a. We fill in the box with one allele from each parent. Then we go back and figure out what this problem is asking. So we're asking, will they yield an albino offspring? So if we look at our box, all of these are big A, little a, heterozygous, and as long as there is a copy of the dominant allele, these giraffes will have pigmentation. So there's a 0% chance of having an albino baby. Not every gene shows complete dominance. We are going to look at two other cases. Incomplete dominance is when the heterozygote appears to be an intermediate between the two homozygotes. So, in these flowers, we see that there are homozygous red flowers and homozygous white flowers. If you mix those two and have a heterozygote, we see that the offspring will be pink. Another case is codominance. This is when the heterozygote displays characteristics of both homozygotes. This means that both alleles are equally dominant and both will show up in the heterozygote. So in this case, with these cows, we have a homozygous white cow, a homozygous brown cow, and in the heterozygote, we see both brown and white spots. Let's practice with incomplete dominance. In this case, we have black coats in dogs are incompletely dominant to yellow coats in the Labrador Retrievers. The intermediate heterozygous condition results in a brown or chocolate lab. So we see this is incomplete dominance because the dominant allele, the black coat, is not completely masking the yellow coat or lack of pigmentation that we see in the homozygous recessive. This results in sort of a blending of the two colors and gives us the chocolate or brown colored lab. This question asks, a brown lab is crossed with a chocolate lab. What is the genotypic and phenotypic ratio of the puppies? To start this problem, we need to make a key of alleles. In this case, we're going to use the letter B. So big B will be black and little b will be yellow. A brown lab is crossed with a chocolate lab. This is two ways of saying the same phenotype. Brown, chocolate, same thing. So we said before that brown means that we have a heterozygous individual. So big B, little b, crossed with a chocolate lab, which is also brown, so another heterozygote. And so now we've got our parents. We're going to put their gametes on a Punnett square. So there's our Punnett square. We've got big B, little b for parent number 1, big B, little b for parent number 2. Now we can fill in our square. So big B, big B, big B, little b, big B, little b, little b, little b. Okay, so the question asks for both the genotypic ratio and the phenotypic ratio. So genotypic ratio is reading the genes. So what are the actual ratios of the letters in the Punnett square? So in this case, we have 1/4 of our babies are big B, 1/2 of them are heterozygotes, big B, little b, and 1/4 of them are homozygous recessive. For phenotypic ratio, we're going to be looking at the actual colors of the dog, so what we see, the physical features. So in this case, we have to translate these big B's, little b's into an actual color.
So Big B is going to be black. 1/4 of our boxes have the Big B, so we have 1/4 black puppies. Half of the boxes have Big B, little b, so those are brown dogs. And the last 1/4 of the boxes are little b, which means that we're going to have yellow labs.
With codominance, we see that 2 alleles are both dominant. Neither one is masked by the other. So in this case, we're talking about dogs that are black and white, and both of these colors are dominant. The first thing we want to do is set up a key of our genotypes. So with both incomplete dominance and codominance, that is a crucial part of our problems. For this problem, we're looking at black dogs and white dogs. So Big B is going to be a black dog. Our heterozygote, Big B, little b, is going to show up with both types of spots, black and white. Lastly, our little b is going to be white. So for this problem, we're looking at a black and white dog, so the heterozygote, crossed with a solid black dog. It's going to be Big B. Once we have our cross, we're going to put our gametes into a Punnett square. So Big B, little b for parent number 1, Big B for parent number 2. We're going to fill those in with the appropriate gametes. Okay, this question is then asking what is the chance of getting a puppy who is solid black. So now we need to look inside of the Punnett squares and see which genotypes correspond to black individuals. So if we look back at our key, we know that Big B is the black puppy. Okay, and there are 2 boxes with Big B. So to answer the question, we have a 50% chance of having puppies who are black. In this video, we have investigated ways to solve different genetics problems. We have looked at complete dominance, incomplete dominance, and codominance.